Okay. So this is a 5-week course. Um, and we will be covering like units 2 and 3. That includes all the bases of biology, like, life processes, cells, and how basically organisms interact with the environment, and also the human body systems, and of course its organelles. Its organs that is very fundamental in our life. And all of these are the characteristics of living things, right? So before we start our lesson, just gonna say some rules for this one. This is a half an hour like course, so you can like go get your snacks. Feel free to go to the bathroom. Just type it in the chat. So yeah, okay. And I want you to like get to know each other. So just tell me your name and what do you call like your nickname. And
My name is Amina, and like people call me Mo most of the time. That's my nickname.
Mo.
Yeah.
Okay. And tell me how do you like prepare for certain exams, any subject?
So I procrastinate a lot,
Oh no,
and
no.
I have like anxiety issues. I go through them all, but I do study, and I like the test got easy for me, but
Mhm.
still I have like anxiety issues and depressions,
Oh no.
all
You
of
have
that.
to
I have
like
to go through
control your
all of them
Mhm.
every time I have an exam.
So you're like you're shaking your hands or like very nervous when you're
Yep,
taking.
all the time.
Exactly, because that's like the usual students what feel during the test. And yeah, so this is me. I'm Genevieve Domingo, and I'm a junior high school in Francis Louis.
Oh,
And yeah, so here is the content of our 5-week course. We will be going over the organelles, life processes, human body system, and homeostasis. So I think you already know some of it, right?
yeah, I did it in eighth grade.
Okay, that's great. So it's like a review for you.
Yep.
So this course it will only be like the half course because like every semester it's a different like type of topics. So we can't go like all the topics in biology. So I'm going to do the half semester, and maybe like the next semester I'm gonna do like half of it, but I'm not sure. So just be updated to it.
Okay.
And here's the little course schedule. If you see this, I posted it.
Yeah, I saw
On
that.
holidays we don't have classes, so yeah. So you can, we're not allowed to go out, so just stay home.
Yeah, I'm not gonna go. I'm scared.
Yeah. And okay. So, okay. Oops. So today is October 10th, right? So we're just gonna talk about the little introduction of organelles, more modern cell theory, and half of the life processes, and a Jeopardy game. Do you want me to like post an activity, or just let's just go over the worksheet that I posted?
Let's just go over the worksheet.
Okay, that's great. Okay. So organelles, okay. So before we discuss the basic unit of life, I want to give out some tips and ideas on how like to ace the living environment regions. Wait, did you already take it or no?
Yeah, so basically I did it in eighth grade, but I couldn't take it because of Corona. But I think I got the credit for it.
Oh, so
They saw my results, and I think I got passed for it.
so
Um,
they just base it in the class?
I'm not sure, but everyone's saying that if you are not taking that class again, that means you got the credit.
Mm, So no exam at all.
Yep.
That's so sad. So I think you already know what are the things that you should do and you should not do.
Mm.
So like always practice your thing, and then there's some websites like Khan Academy or Civum.com that you can go into. I'm gonna send the link, and
Okay.
yeah, that would be all for that. Hmm. So as I mentioned earlier, organelles are the basic unit of life, which mean every organs that are working in the human body system are powered by cells. And when we study science, we tend to categorize that cells, atoms, molecules, and proteins are very small. But we don't think, like some other people think that, oh, this is similar because you can even see it with your naked eye. So, but for scientists, uh, it's a huge difference. The sizes and how they function and the structure. Okay.
Is there any connection between cells and atoms and molecules?
So basically, we're talking about the scale, like the how big is the cells. But I'm not gonna go deeper into it because yeah, this is just like a fun course.
Yeah.
We're not scientists here. We're just high schoolers. Okay, so I wanna ask you a question, like critical thinking. So your brain is starting to like think.
Okay.
So, what do you think would happen if one of the part in organelles like failed to do its job?
I think there will be problem in homeostasis because like if one cell is having problem, maybe like other cells are having the same issues, and like cells matters because your energy comes from there. The carbon dioxide and oxygen, all of these things are happening in there, so it's important.
Exactly. So, I also want to add like if we don't have like the nucleus in our cells, because the nucleus is
Yeah,
like the leader.
genes,
So,
genetic.
if you don't have that, so there's no leader at all, you're
Yeah,
dead. Okay. So, we can like create like analogies in the school. The principal is like acting the nucleus, and if we don't have the principal in our school, you know, the school will be
yeah.
chaotic.
It won't like get the instruction correctly.
Exactly. So that's why that's the importance of the nucleus. And now let's get to the modern cell theory. Okay. Do you want me to read it or you just want to read this?
I can really.
Okay. Just read the first bullet and I'll explain it.
All living things are made of cell.
Okay. So you already know that cells provides us structure, energy. That's all for the body, right? And that is why we are like firmly formed as a human or human being. And same thing with the other living things. The second bullet point, I'll read it for you. The cell is the smallest living unit in all organisms. So it basically means that it's the building blocks of life. So like the other molecules, they have like the building blocks of um can you think like any other molecules that have the building blocks or blank?
Like molecules?
Yeah, like starch, glucose, they have like the building blocks of
Oh, I can. My brain is. I don't know.
That's fine.
For
It's
some
okay.
reason, I can't remember, but I know them.
That's okay. Okay. So the third bullet point is that all cells come from other pre-existing cells. What do you think about it? Like any
Like
idea?
cells divides and then it makes two other new cells.
Yeah, this is mitosis and meiosis. And
Yeah.
for that one, we will talk about that like in the future.
Okay.
And so, can you guess how many cells are in a human body?
Millions and trillions, maybe gazillions.
We don't ever know.
Yeah, like a lot.
Yeah, it's really a lot. Okay. Mhm. So on the screen, you'll see the picture of prokaryotic cell and eukaryotic cells. And I want you to talk, I want you to like compare and contrast, like what are the difference and similarities between them?
Okay, so like the first one that looks like a sperm. I don't know why. Is it?
No, it's a bacteria.
Oh, okay, it's a bacteria. Okay, so
Yeah.
there's nucleotide. Okay, and then cell.
So
Doesn't
what
have
we,
mitochondria in it.
yeah, they
Another
don't
one
have.
has.
Mhm.
Yeah, and
What else? The main part that we talked about earlier.
I forgot.
Starts with N?
Nucleus.
Yeah. So prokaryotes don't have nucleus, but they have their own genetic material. Same thing with eukaryotes. They have their own genetic material to produce and repair. And that's okay. What else do you see?
It doesn't have cytoplasm.
Cytoplasm.
Yeah, does it? The prokaryotic cell.
Oh yeah, because it doesn't hold the organelles itself, right?
It doesn't hold organelles itself.
Wait, is that what you think, or for
But
prokaryotes?
something is holding it together, but I don't know.
They do have cytoplasm for prokaryotes because those nucleoids are the DNA, so it keeps on the like in place. So they do have cytoplasm.
Oh, the master changes the flagellum or something. That word.
Yeah,
The
flagellum.
tail.
Yeah, that's the tail for their movement.
Oh, yeah. And the eukaryotes does
Oh,
not have
so
that.
that's why you thought it was a sperm.
Yeah.
Okay. So we're ready compare and contrast, and I'll show you a short video. Okay. Can you hear it? Okay.
On Earth, there are two major types of cells, prokaryotic and eukaryotic. Prokaryotic cells are the simplest and most ancient types of cells. They were the only form of life on Earth for billions of years before eventually giving rise to eukaryotic cells. If you take a look at prokaryotic and eukaryotic cells side by side, you can see a number of differences. To begin with, prokaryotic cells are a lot smaller than eukaryotic cells and have a simpler structure. But bear in mind that this simple structure is actually a good thing for prokaryotes because it allows them to reproduce very quickly and very effectively. If you were to look inside a prokaryotic cell, you would probably be surprised at how simple it is. For example, prokaryotic cells lack a nucleus. In fact, the name prokaryote actually gives you a hint about its structure. Kary means kernel or nucleus, and pro means before. So basically,
Okay. So I want to stop right there because the way they have the clue in its name is kind of complex. So I'm just gonna add like the word pro. It sounds like no, right? So you already know that it doesn't have nucleus, so no nucleus, and we are also going to talk about that they don't have membrane-bound cell organelles, right?
What's the difference between nucleus and nucleotide?
Nucleotide is you see this like spaghetti
Mm-hmm.
DNA, those are the nuclei. So basically, if you like zoom it in, those are the like little chromosomes, the
Yeah.
making the genes.
Mm-hmm.
So yeah.
Those are nucleus.
No, 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 no. That's not the nucleus. That's the nucleolide.
Nucleotide.
Yeah, you get it.
Basically, the word prokaryote means before nucleus. The inside of the prokaryotic cell is basically an open unit with no compartments. There are no membrane-bound organelles. However, small structures called ribosomes are scattered throughout its cytoplasm. The cell's DNA is located in a region of the cytoplasm called the nucleoid region. The nucleoid region is not the same thing as a nucleus because it's not enclosed by a membrane. Prokaryotes also have a cell wall that surrounds the plasma membrane. The composition of the cell wall depends on whether the cell is an archaean or a bacteria, but basically it consists of complex polysaccharides. Prokaryotes also often possess one or more flagella, which are used for movement. In contrast, eukaryotic cells contain a membrane-bound nucleus and other membrane-bound organelles. The name eukaryote also gives a hint about its structure. Eu means true, and remember that karyote means nucleus. So in essence, the word eukaryote means true nucleus.
Okay, so I'm gonna add something also that from the word "you," it sounds like "do." So they do have membrane-bound organelles. You see this mitochondria. They have the covering itself. Also the nucleus. They have like a little home, and then same thing with the cell itself, right?
Okay.
So basically, this means that these organelles are semi-permeable, or it allows the substances or molecules to go across the wall.
Yeah, like carbon dioxide and
Yeah,
oxygen.
yeah. So give me like an example that molecules cannot go in, you know?
Like what molecules cannot
Yeah,
go in?
what molecules?
Um, molecules that are bigger than this. So what about like glucose?
Yeah, glucose. Yeah, that's what I'm finding. Okay.
Eukaryotic cells are larger and much more complicated than prokaryotic cells. However, they do share a few things in common with prokaryotes. Let's take a look at a Venn diagram to compare prokaryotes to eukaryotes.
Oh, you can copy this if you want, because it's a
Okay.
good reference. So yeah.
We'll also see what areas they have in common. Let's start with their commonalities. Both prokaryotes and eukaryotes have DNA as their genetic material, have ribosomes, have cytoplasm, and have a plasma membrane. Now let's look at some of the ways they differ. Prokaryotic cells are the oldest type of cell. They're small and relatively simple. Eukaryotic cells evolved from prokaryotic cells later. They're larger and much more complex internally. Prokaryotes lack both a nucleus and membrane-bound organelles. Eukaryotic cells have a nucleus and also contain organelles. Prokaryotes are single-celled organisms. Eukaryotes can be either single-celled or multicellular. And finally, the DNA of a prokaryote is usually organized as a single circular chromosome, while the DNA of a eukaryote is organized as linear chromosomes.
Okay. Let's end it right there and go back. Oops, sorry. Okay, so since we already know that prokaryotes are in simple organization and can like independently live by itself, such as like bacteria, protists, and fungi, right? Or fungi, the way you, whichever you will. Yeah. So while the eukaryotes, on the other hand, they are more complex. So that is why they're called multicellular, right? And let's move on. Here are the two types of eukaryotic cells. This is like the main, like the usual that you will see in your class, the animal cell and the plant cell. And on this photo, it includes like a lot of details, but we're not gonna like go into that depth part because we're gonna pick the most important organelles such as mitochondria, the nucleus, the nucleolus, you know, the usual.
Yeah, like the important ones.
Yeah, because they have like different structure that it's not like essential to it. So yeah. So can you give me like the difference and similarities, like the same thing with the eukaryotes and prokaryotes?
Wait, aren't this like animal cell and a plant cell?
Yeah, the animal cell is on your left and the plant cell is on your right.
Yeah.
So any
So what did you tell me to say? Sorry.
Compare it. It's the same thing. Compare and contrast.
Compare it with the prokaryote one?
Oh no, plant cell compared to animal cell.
Okay, so the animal cell does not have a cell wall.
Mm-hmm.
And then the plant cell does not have like a nuclear pore.
Yeah.
And okay.
The most
There's like
important
wait.
Wait,
I
what?
did do this in 8th grade though.
You did? So you already know about this.
I literally forgot everything. Oh my god.
So you need like a refreshment. Okay, so the cell wall is the
Yeah.
important for the plant cell, which is not, it doesn't exist in animal cell, right?
Animals.
Mm-hmm.
Yeah.
And also for plant cell, they have like a big vacuole, like
Oh yeah,
compartment.
vacuole.
Yeah. So they
I think
store all
like animal cell has it too, but like small.
small, yeah, very, very small vacuoles that exist in animal cell. So, oops. Okay, I'm gonna show like a little tour inside the cell. So this video will talk about the functions of organelles, and I also posted the notes about the organelles and its structures, so we can like take a look at it later. But right now we're gonna watch a little video.
Now, to start our trip, we're going to have to get through this cell membrane, also called a plasma membrane. It's selectively permeable, which means that it only lets certain select materials in and out, but by doing so, it keeps things in the cell stable, also known as keeping homeostasis. And we have an entire video on just the membrane itself, which is found in all cells. But for now, we're just going to have to squeeze through this protein in the membrane. Now, inside the cell, we find ourselves in this jelly-like material called cytoplasm. It surrounds all these internal cell structures, and you'll find it inside both prokaryotes and eukaryotes. Now, organelles that are floating around in the cytoplasm can have more support than you might think. Cells contain a cytoskeleton, which is a collection of fibers that can provide support for the cell and its organelles. And the cytoskeleton can even play a major role in cell movement. The cytoskeleton actually deserves its own video because it's very complex and its organization can vary depending on what kind of cell you're looking at.
So do you remember what cytoskeleton are?
What?
Cytoskeleton?
These are the like things that holds all the organelles together.
Yeah, like so they don't like scattered a
Move
lot.
away.
Okay, yeah.
Moving through the cytoplasm, let's start with ribosomes. They are not membrane-bound organelles, and they're going to be in both prokaryotes and eukaryotes. They make protein, which is really important because that's what so much of genetic material, DNA, codes for, protein. Ribosomes can be free in the cytoplasm, but they can be attached to another organelle too, which...
What do you think that the ribosomes can be attached on? What type of organelle?
Wait, you just...
Wait, I'm sorry? I think you froze. Okay, I'm gonna type it in the chat. I can't hear you. Hello? Hello?
having the
You're breaking out. Hello? Try getting in again. So let's see. Hello?
Yeah, sorry, I was just having internet problem.
It's okay. Take your time. Is it okay right now?
Yeah, it's okay now.
Okay, so what I was saying before, that the what type of organelle that the ribosomes are attached at. You can type it in the chat. I can read it.
What type of organelles are ribosomes attached to?
Yeah.
Um,
I'll give you a clue. It starts with like the shortcut for that word is ER.
endoplasmic reticulum?
Yep. You're right.
We'll talk about a little bit later. We're now going to focus on organelles that will be membrane bound. So we're going to be focusing on organelles that you would find in a eukaryote cell. This takes our travel to the big boss, the nucleus. Now in eukaryotes, it holds the genetic material. Genetic material as in DNA, for example. Both prokaryotes and eukaryotes have DNA, but if you're a eukaryote, you have a nucleus to put it in. And the nucleus controls the cell's activities, and inside it, it has a nucleolus, which is where ribosomes can be produced. Attached to the membrane of the nucleus, or nuclear membrane, you will find the endoplasmic reticulum, ER for short. It does a lot of processing of molecules for the cell, like protein folding, and it's highly involved in actually transporting those molecules around, like a highway. There's a rough ER, which has ribosomes attached to it, making it, as you can imagine, rough, and then smooth ER, which doesn't have the ribosomes. Rough ER specifically tends to be involved with protein producing and transporting, because remember that ribosomes make protein. Now, molecules that leave the ER can be sent away in vesicles that actually pinch off the ER themselves. Now, smooth ER has many additional roles, including detoxification, which is one reason why your liver cells tend to have a lot of smooth ER. And another additional role of smooth ER is that it can make some types of lipids. Next, the Golgi apparatus. It's the ultimate packaging center. It can receive items from the transport vesicles that pinched off the ER. It has enzymes that can modify molecules it may receive and sorts the material it receives as well. It can determine where to send those molecules, including some that may eventually be sent to the membrane, so they can be secreted, which means items that can be sent out of the cell. So with all that's going on in here, you might start to wonder, what's powering this thing? The mighty mitochondria, or mitochondrion, if you're just talking about one. They're like power plants. The mitochondria make ATP energy in a process called cellular respiration. Now, it's not the type of power plant that you might think of. It runs on glucose, which is a sugar, and it needs the presence of oxygen to efficiently make ATP energy. Now, at this point, we need to mention that eukaryotes are not a one-size-fits-all. Animal cells can have differences from plant cells. We have a fork in the road here. For example, Plant cells not only have mitochondria, but they can also have these awesome organelles called chloroplasts. Chloroplasts actually make glucose by using light energy in a process known as photosynthesis. They tend to even have a green look to them because they have a pigment that captures the light energy and it reflects green light. Now, both plant and animal cells can have vacuoles. Now, vacuoles can have a lot of different functions, but many types act as storage of materials. Plant cells can have one large vacuole called a central vacuole, while animal cells can have smaller several vacuoles. Now, remember how we already said that all cells have membranes? They do. But plant cells additionally can have a cell wall, which is a layer that offers additional protection and shape maintenance that animal cells do not. Okay, now how do we get out of this animal cell that we've been in? Well, we could get out like a protein would. So if we were a protein, we would only be made because of instructions from DNA. And remember that in eukaryotes, DNA is found in the nucleus. We would be made by a ribosome because ribosomes make protein. The ribosomes could be attached to the rough ER, and the rough ER highway would provide a vesicle to send us to the Golgi apparatus where sorting can take place. And if we're tagged for being secreted, we could be sent off through a vesicle from the Golgi to the membrane. Out we go! Now just keep in mind that in our quick tour, there are still so many more awesome organelles found in lots of different types of eukaryote cells to continue exploring, so to the Google for more.
Okay, so for lysosome, do you have any idea what is that?
No.
Okay, so basically lysosomes are, you know, when
Cleans
this,
out. It cleans things. Is it?
Yes.
yeah, it engulfs the intruders, or like the thing that makes the cell fail to function.
Mm-hmm.
So if it triggers the lysosome, it will eat the cell itself. So
So it
it's
will
gone.
clear it out.
Yeah.
Yep.
hmm I think you got this.
I pretty much got the idea.
Yeah. Okay. So just a little like remember here, the cells in the human body works together as a whole to function all the organ system, and the cells are independent. They can survive on their own, you know, because of their specific function. So even the prokaryotes can live like thousands of years, millions of years. Same thing with other types of cells. And yeah, let's go here. So you know, microscope can be used to see cells, and that's usually can be seen in science lab. Have you ever used like a microscope?
Yeah, I did use like a microscope, but not that. Like I haven't seen a cell.
Oh, you haven't tried it?
No, I did use the microscope, but I haven't like looked at a cell, like a living cell.
Oh, so that'll be interesting for you in the future.
Like, what does it look though?
You want me to search it out for you? Because it likes
No,
it
I
has
will.
like
I will like see it when I do the research.
Okay. So basically, you said earlier that you don't want to play the game because it's just the two of us, and I'll just show you the worksheets that I sent out. So basically, for the worksheets that I sent or posted, it's not mandatory because I'm not gonna grade it. We're just gonna check it.
It's just for like our practice.
Yeah, it's just for practice. And if you need help, like you're stuck or you're having trouble with that certain question, just email me. I'm always free.
Sure.
Okay. Do you see it?
Yeah, I think I did this worksheet in 8th grade.
Exactly the same thing?
Yep. It was in my test,
Oh.
but I couldn't remember any of them. Though.
Okay, so we can like go
Yeah,
over.
we can go over it.
Okay. So let's start with number 1 on this side, on your left side. So maybe we can do like 10 questions.
Yeah, sure.
So first, which structures could most likely be observed in cells in the lower power field of a compound light microscope? What do you think?
I think cell walls and chloroplast, because these are like the out, like the
Yeah, exactly.
really okay.
Mhm. So that one is right. Second, which organelles must be present within a cell of a geranium leaf for respiration and photosynthesis to occur?
I think that's mitochondria and chloroplast.
Exactly, because for
So like basically, since we only have mitochondria and like a tree has mitochondria and chloroplast, does that mean like tree can function more than a human?
wait, say it again.
So basically, human cells only have like mitochondria, but tree and plant cells has mitochondria and chloroplast. Does that mean plants are like more better than human cells?
It can really say it's much better because plants are autotrophs and they can make their
Yeah,
own,
make their own food.
but us we only consume them. So I would say some kinda, yeah,
So like plants are more powerful than humans.
yeah, I think so. And number 3, in the diagram of a cell below, the structure labeled X, the big, like, circle.
At the vacuole or
Yep. So what
okay,
does the vacuole do?
store water. Store
Yep. Store water. Exactly. So number 4, if the ribosomes of a cell were destroyed, what effect would this most likely have on the cell?
Couldn't make protein. Synthesize the protein.
Yep. I think you got this, right?
Sort of.
Sort of. So we can continue, or you want to go to our slides?
Other slides, I guess.
Okay. I'll just leave it here. Okay, so life processes. Here we go. Can you see it?
Yep.
Okay. So here are the life processes. In your class, you may already know about Mrs. Nerg, but in my past living environment class, we use this regions acronym. So in your past class, did you use this acronym, Mrs. Nerg?
No, we didn't use any acronym,
For
but I
life?
do remember all the terms.
Okay, so the word regions, it's it's easily to know because you're taking regions.
Regions,
so,
yeah.
Okay, so let's talk about the respiration first. Do you have any idea what is
Taking breathe, like oxygen and carbon dioxide, the whole process is respiration.
Yeah, but what is happening in that certain
So
process?
we're like inhaling oxygen and it is going to our cells, and then like the cell is taking all the energy from the oxygen and then like somehow pass the carbon dioxide and the carbon dioxide go out.
Great.
And then ATP is there too. ATP and
Yeah,
mitochondria.
ATP is there. And there's 2 types of respiration, anaerobic and aerobic. So basically aerobic, from its word air, they do have air and they require oxygen. Okay. And the reactants that involved are glucose and oxygen. So its formula equation, I sent it on the notes over here. I'm gonna show it to you here so you know what I'm talking about. This is the formula or the combustion, how the glucose and oxygen are being used, and then it will release carbon dioxide and water.
Okay.
So let's back. Sorry. And regulation. So far you know what the respiration is, right?
Yep.
So can you give me like an example?
Of like respiration.
Yeah. Other than like in our activity, human activity.
Please. I only can remember of taking breathe and.
So what does the breathing? Okay, so we can say like when you're jogging, right?
Mm-hmm.
So as your heart rate and your breathing rate increases,
Yeah.
the lactic acid builds up in your muscle. So it means you are lacking of air. This means this is anaerobic.
Oh, sounds complicated.
Yeah, it sounds complicated.
But I do get it, sort of.
So yeah. But you recognize what is lactic acid, right?
Yep.
Okay, so let's move on to regulation. Regulation is all activities that involve maintaining homeostasis, like you said earlier, and then it is the process of where living things are being controlled, like in and out. So the best example for this one is the guard cells in the plants. So you know, like the stomata, like the opening and the closing of the cells.
Yeah.
So it controls the water coming in and coming out of the leaves, right?
Yep.
Okay. And what else? Some of examples we can also say about the nervous system. You know, like the nerve cells, it also controls like our thinking. And
Okay.
let's go for reproduction. So you already know what is reproduction is, right? So it produces new organisms, like we said
Like
earlier.
mitosis and meiosis, all those steps. Yeah.
Yep. So we also have two types of reproduction. Asexual reproduction, which
Like
requires
bacteria.
Yep, exactly. And sexual reproduction, you know
Like
what?
humans and.
Yep. Okay. So growth. So basically for growth is like you are building up or
Multiple
developing.
cells.
So we can say that your proteins are giving you to like repair your hair, your keratin, so your hair gets longer, your skin, the changes of your color, you know, like the melanin.
Yeah.
Okay.
I'm sorry.
It's fine. You can type in the chat if you don't want to unmute yourself because I can see it.
Yeah, sure.
Yeah. So yeah. Excretion. So for excretion, we're talking about how the like you know when we were like jogging around or exercising our sweat, right?
So like sweating and
Sweating,
like,
yeah.
Like all the way like our waste product goes out.
Mm-hmm. And that's easy. Nutrition, you know what is the difference between autotrophs and heterotrophs.
Heterotrophs, yeah.
Yeah. So are we good with that?
Yep.
Okay. So transport, this is the main one that we're going to talk about. This is the absorption of materials and movement or circulation of materials throughout the living organism. So over here we have active transport and passive transport. And we're gonna talk about the active transport first. So the movement of molecules goes from low to higher concentration, and it uses ATP or energy. So over here, I'll show it to you. Oops. Can you see it?
Yes.
Okay, so active transport, as we already said, it uses energy. It also requires, do you see this oval shape yellow like, this is the channel protein, which we are gonna talk about later on. And
That is a protein.
yeah, channel protein, yeah.
Okay.
So the molecules that are like going across the membrane will go inside through this channel protein.
And this is like
Hmm?
the membrane.
Yeah, this is the zoom in picture of the membrane. Okay, so endocytosis and exocytosis. I'm sorry, this is so hard to read.
Vowel is like the name is more hard.
So endocytosis is the way the membrane engulfs the molecules outside. So basically what they do is just they take the particle outside and then heat. And the exocytosis is the opposite. So it just releases from the word exit and from the word endo, in, right? Do you recognize this too? Endocytosis and exocytosis?
Yeah, so like on endocytosis, things go in and exo go out.
Yep. And we're also going to talk about the hypotonic or the swelling stage. If you check it, this is hypertonic. This is how the water are being released and being accumulated by this is red blood cell and the plant cell. Okay. So you see the water is coming out, so it means the red blood cells are shriveled or
Doesn't that happen by diffusion?
diffusion, or I
Like
think
active transport?
not diffusion. Remember, we're talking about water.
Yeah.
So this is osmosis.
Oh yeah. Osmosis is for the water one. I got it.
Yep. So let's go back. So passive transport, the movement of molecules from high to low concentration, and this doesn't require ATP. And diffusion and osmosis, you know, the movement of water and how the molecules go into the cell. That's diffusion. And hypertonic is the shrinking stage. This one.
Yeah.
So over here, when you see like the red blood cells are popping out, it means it overloaded or excess of water coming in. And isotonic is the normal one. So this is like the usual or the common. So I'm gonna put this video.
Cells are really picky. They're always either too hot, too cold, too salty, or too watery. In order to survive, cells need to maintain a specific internal environment. They're constantly adjusting the amounts of glucose, water, oxygen, carbon dioxide, sodium, potassium, calcium, and many other things. But how do cells do this? How do they make sure that they maintain a perfect internal environment? Stick with us, we're going to find out. The process of constantly adjusting certain variables is known as dynamic homeostasis. Usually it involves feedback mechanisms called negative feedback loops. Cells use both passive transport and active transport to move materials in and out. However, the size and polarity of the molecules will determine how they can get across the membrane. Remember that cells are surrounded by a double membrane made of phospholipids and cholesterol. This barrier is excellent at stopping most molecules from passing through while still allowing certain particles to cross. As we go through the different types of particle transport, we'll be keeping our information organized in a grid that looks like this. We'll be starting off with passive transport and a process called diffusion. In diffusion, particles flow from an area of higher concentration to an area of lower concentration. The particles spread out until they're equally spaced apart. Diffusion becomes much more complex when we add a membrane. The particles still want to flow from high concentration to low concentration, or as we call it, down the concentration gradient, but the membrane gets in the way. Luckily, small and nonpolar molecules can slip right through the membrane. These include both oxygen and carbon dioxide. To summarize, in diffusion, the particles only flow from high to low concentration, do not use protein channels, and also do not need to spend any ATP to do it. Next up is a slightly different type of passive transport called facilitated diffusion. Remember that large or polar molecules can't fit through the cell membrane. Water isn't a very large molecule, but its polar structure makes it unable to slip through the membrane on its own. In facilitated diffusion, the cell uses channel proteins to allow...
Oh, this one, I said this earlier, the orange one. This is
The
a channel
protein.
protein. Yeah. So the other
large
cells
and or
or the
polar
other
molecules to
molecules
pass through.
can
Water
pass through.
diffuses through the cell membrane using special channel proteins called aquaporins. Whenever water diffuses across a membrane, we call it osmosis. To continue with our chart, in facilitated diffusion, particles still flow from high to low concentration. They do use protein channels, but they still don't use ATP. Next, we're going to move into the realm of active transport. Active transport requires the input of energy, usually in the form of ATP. Remember that ATP, or adenosine triphosphate, is a rechargeable, renewable source of cellular energy, almost like a battery. It stores energy in its bonds, and that energy is released when the bonds are broken. The ATP is reduced to lower energy ADP, then is recharged into ATP via cellular respiration.
So in living environment, ATP is the most important one. So we kind of don't more focus on ADP. So yeah.
Active transport often uses channel proteins, similar to those used in facilitated diffusion. These channel proteins charge a toll to admit a molecule. In return for a small amount of ATP, these carrier proteins allow certain molecules to pass through the cell membrane. These carrier proteins are especially useful because they can pump items against their concentration gradients. Passive transport can only allow particles to flow from high concentration to low concentration, but active transport can pump from low to high, thereby making the concentration more extreme rather than only moving towards equilibrium. A great example of this is the sodium-potassium pump, which pumps sodium out of cells and potassium into cells, allowing neurons to work. 3 sodium ions enter the carrier protein, which is then charged by a molecule of ATP in a process called phosphorylation. The
This is
protein
more
then
complex,
undergoes a
so
conformational change,
we're not gonna
delivering the
focus on
sodium
this.
to the outside of the membrane. The process is then reversed, this time delivering potassium to the inside and returning the protein to its original shape. In active transport, it's possible to pump particles from low to high concentration as well as from high to low. The process uses protein channels, and the cell has to spend some ATP. Sometimes, however, it's not efficient enough to transport molecules through the membrane just a few at a time, so cells use a process called bulk transport. There are 2 major types of bulk transport, and they're related. They're called endocytosis and exocytosis. Neurotransmitters are a great example of bulk transport. These molecules allow neurons to communicate. Just like other cells, neurons are surrounded by a phospholipid bilayer. Neurotransmitters are encased in vesicles that are made of the same material. These vesicles are brought to the edge of the presynaptic neuron. The vesicles dock against the membrane and then release their contents into the synaptic cleft between the 2 neurons. The vesicle is then incorporated into the neuron's cell membrane. The presynaptic neuron has just performed exocytosis. When the particles of neurotransmitter lock into specific ligands on the postsynaptic neuron, endocytosis can occur. The particles...
Okay, so these different shapes are called receptor molecules, where the particles or the vesicles that came out from the other neuro are being completely fit to that particular shape.
It's from
So
enzyme, right?
yeah, it's like this enzyme, and we can like compare it, like when you're charging your phone, it fits exactly to your, like, let's say iPhone. So the connector of that phone only fits to iPhone, not Android. So it's the same like analogy.
are again wrapped up in a bubble of cell membrane that becomes a new vesicle. Endo and exocytosis are capable of moving particles both with and against the concentration gradient. Instead of using protein channels, they use vesicles to transport particles. The vesicles can hold more molecules, but opening and closing them requires energy, so the cell does need to spend a little bit of ATP. So there we have it, our grid that compares and contrasts passive and active transport. If you'd like to learn more about homeostasis,
Let's move on with that. Do you want me to show what's inside the structure of the cell membrane? So you have like any idea what's...
I don't know if you want to.
Okay, let's just go over it.
Welcome back, everyone. For this week's video, we're going to be discussing the cell membrane, specifically the fluid mosaic model and what makes it semi-permeable. Okay, here we go. The cell membrane has several major roles. It protects the cell from its surroundings, maintains an internal environment that is different from its external environment, and determines which particles may enter or exit the cell. It has many components that allow it to perform all of these functions. These include phospholipids, cholesterol, channel proteins, carrier proteins, glycoproteins, and peripheral proteins. The membrane they create is both fluid and selectively permeable.
Okay, let's go back. So the important ones that we're gonna talk about later on is phospholipids, channel protein, and the cholesterol. We're not gonna focus on this glycoprotein and peripheral protein because those are like really complex for biology.
Okay.
And selectively permeable. Here's a picture of the cell membrane. Notice how it has two layers. These layers are comprised of what we call phospholipids. The phosphate head is both polar and hydrophilic. Attached to it are two non-polar hydrophobic fatty acid tails. These tails can be either saturated or unsaturated.
Okay, so from the word hydrophilic and this head, it loves water. But on the other hand, the hydrophobic, from the word phobic, it doesn't want to be exposed to water. Yeah.
Phosphate groups have a charge and the tails do not. The nonpolar tails face inwards towards one another and the polar phosphate groups are on the outside. This arrangement keeps molecules from passing through too easily. In between every few phospholipids is a molecule you've probably heard of, cholesterol. Despite what you might see in all those pharmaceutical commercials, cholesterol is actually really important. It helps the cell membrane maintain an appropriate level of fluidity by managing the space between phospholipids. At room temperature, the cell membrane has about the same consistency as vegetable oil, which is ideal. However, higher temperatures would cause the cell membrane components to move farther away from each other and the membrane would disintegrate. Conversely, colder temperatures would cause the phospholipids to clump together and make the cell membrane too rigid to work properly. Cholesterol has a very stable structure and it helps the phospholipids stay at the optimal distance from one another, almost like an overzealous chaperone at a middle school dance. So how exactly does the cell membrane control what molecules can pass in and out? Certain molecules, like oxygen and carbon dioxide, can easily squeeze through it without any special accommodations. This is lucky because we exchange a lot of oxygen and carbon dioxide during cellular respiration. These molecules are small and uncharged, so they can sneak through using just diffusion. However, other molecules can't get through without help, and this is where many of our proteins are going to come into play. Molecules that are large or charged can't get through the fatty inner layer. Specialized channel proteins allow large or charged molecules to pass through the membrane. These molecules flow down the concentration gradient from high concentration to low concentration. Structures called carrier proteins charge a toll to admit a molecule. In return for a small amount of ATP, these proteins allow certain molecules to pass through. They are especially useful because they can often pump items against their concentration gradients. A great example of this is the sodium-potassium pump, which pumps sodium out of cells and potassium into cells, allowing neurons to work. The cell membrane also often includes glycoproteins, which have a protein component embedded in the cell wall, and then a carbohydrate protrusion that dangles out away from it. Antibodies involve glycoprotein usage, but these proteins also play a major role in conception. Human egg cells are surrounded by layers, including the corona radiata and the zona pellucida. Sperm cells contain special enzymes in a compartment called the acrocell.
I know.
These enzymes break through the corona radiata, exposing the glycoproteins in the inner zona pellucida. The sperm then attaches itself to these glycoproteins, which begins the process of fusing the two cell membranes so the genetic material can be combined. Finally, cell membranes are also studded with
So this
peripheral
is the
proteins on one side of the
receptor
membrane.
molecule,
They
the
can attach and remove
glycoprotein.
themselves from either side of the cell membrane over and over again. You've probably come across a number of peripheral proteins if you've studied cellular respiration. The compartments of mitochondria are bound by a phospholipid membrane, which is studded with proteins, just like the ones earlier in this video. Cytochrome C is an important protein in the electron transport chain, the final steps of cellular respiration. Interestingly, enzymes associated with the cytochrome C protein are one of the targets of the poison cyanide. To wrap up, let's consider this idea called the fluid mosaic model. We've learned that the cell membrane is fluid thanks to cholesterol, but what about this mosaic idea? A mosaic is a piece of art, usually on a wall or a floor, that's made up of small pieces of pottery, stone, or tile. The pieces are usually different colors and arranged in a pattern or to form a picture. For this video, we've been looking at the membrane from the side, but if we flip it around and view it from the top, it does look a lot like a mosaic. All right, that's it for this week.
So mosaic can be also seen in churches, like different colors of figure images.
Yep.
The mosaic can be seen in churches or like any, in museum. So you have like the reference about it. So this is the tonicity about the movement of water going in or out. And let's talk about it.
Imagine that you're stranded on a raft in the middle of the ocean. You don't have any emergency food or water, but you also know that humans can only survive about 3 days without fluids. Should you drink seawater or not? Stick with us. We're going to find out and also review hypertonic, hypotonic, and isotonic solutions along the way. Cells have a very finite range of conditions in which they can survive. If it's too hot, too cold, too watery, too salty, too acidic, or too basic, they can't function properly. We're going to explore what happens when you place a cell in a solution that is much more salty, hypertonic, much more watery, hypotonic, or has an equal salt concentration, isotonic. Remember that cell membranes are selectively permeable. They allow certain particles to pass through, but not others. Usually, larger molecules can't fit through the membrane without special channels. The cell membrane is fitted with special protein channels called aquaporins, which allow water molecules to pass through without expending energy. However, the ease with which the water molecules can cross is going to depend on something called the concentration. Concentration is a measure of how much solute there is per volume of solvent. In other words, whether the liquid is more salty or more watery. We'd usually expect that the molecules would follow a process called diffusion. They would flow from areas of higher to lower concentration. Eventually, they will reach equilibrium, where there will be an equal concentration on both sides of the membrane. However, in the case of a liquid like salt water, the salt molecules are much, much too big to fit through the membrane, so the only particle that can move is water. Let's look a little more closely. Water molecules are highly attracted to salt molecules. They cluster around salt and really, really don't want to move. The positive parts of the water molecule stick to the negative parts of the sodium chloride, and vice versa. The interactions between oxygen and sodium and between hydrogen and chlorine are called ion-dipole interactions, which you might remember from chemistry.
So that would be discussed like in the future if you take chemistry. Okay.
Hydrogen and chlorine are called ion-dipole interactions, which you might remember from chemistry. Water molecules that are not stuck to a molecule of salt are far more likely to relocate. Today we're going to immerse a cell in 3 kinds of liquids: a very solute-rich liquid, a very watery liquid, and a liquid that's in between. Then we'll see what happens to the water molecules. In this first scenario, the liquid surrounding the cell has many molecules of solute in it. This liquid is hypertonic compared to the cell. It has a much higher concentration of solute particles and a much lower concentration of water. Unfortunately, the solute molecules can't pass through the membrane to reach equilibrium, but water molecules can. The molecules of water on the outside of the cell are going to be obstructed from passing through by the many molecules of solvent. However, the molecules on the inside have much less solvent getting in the way. Water will begin rushing out of the cell. Some water will come in, but the net movement, the overall movement, of the water molecules will be outwards and the cell will shrink. We call this cell contraction plasmolysis. This is also how pickles are made. Now let's immerse a cell in a much less concentrated solution, a hypotonic solution. Compared to the cell, it's much less salty. Some water molecules will exit the cell, but even more will rush in because they aren't obstructed by solute particles. The cell grows in size, and it may even undergo cytolysis and burst. Placing a cell in an isotonic solution, where the concentrations inside and outside are equal, is much more pleasant. Equal amounts of water molecules continue to pass in and out of the cell. The system is in dynamic equilibrium. Particles are continuing to move, but the net movement is zero.
Okay, so I'm just gonna wrap this up because this is way too long. So hypertonic is when the cell is shrinking or getting smaller, and hypotonic is when the cell is getting larger because of the accumulation of water coming into the cell. So sometimes it tends to pop out, and isotonic is the normal because it has the equal amount or concentration of water. So that would be all for that tonicity. And we will be talking about the organization of life next week. So be prepared for that. And you want to play this little quizzes time? Okay. So can you go onto it or no?
Yeah, sure. Wait.
So go to the slides and it's number 17. Or please, oh wait, I can start a live quiz.
Yeah, with the code.
Yeah, that will be easier.
Okay, wait.
Yeah. Are you done?
Yeah, I'm sort of like one question left.
Okay.
I'm done now. Like
Okay.
10 correct and 5 incorrect.
Let's go over it. You want me to explain it? The ones that you got incorrect?
If you want to.
But do you understand it somehow or no?
Some of them I understand it when they give the answers.
I think you're like doing it like fast because of the time, I guess.
Yeah, that time was going away.
Okay. So I don't have to explain it, or there's certain questions that you wanna go over with?
I think I don't have to go over it. I sort of got it.
Are you sure?
Yeah, sort of.
Okay, let's just try to talk about it. So you'll see in the picture there is cells from skin, blood cells, cells from lining bladder, cells from lining trachea. So these groups of cells represent different tissues, right?
Yeah, because there's like a group of cells.
Together, yeah, correct. Which one did you answer? Organelles?
Yeah, I was rushing.
That's fine. Okay. So this one is a little bit longer. So an investigation was set up to study the movement of water through a membrane. The results are shown in the diagram at the right or left. Instead, based on these results, which statement correctly predicts what will happen to the red blood cells when they are placed in a beaker containing water solution in which the salt concentration is much higher than the salt concentration in the red blood cell. So you pick the red blood cells will first lose water, then
Yeah, and I'm
absorb.
absolutely wrong. I could understand.
Yeah. You want me to explain it?
Yeah.
Okay, so basically, they're asking about um. So if the red blood cells are in the water solution, which the salt concentration is much higher, so this means the red blood cell will shrink or get smaller, so it loses water and decreases in size.
Yeah.
You will see it over here, the third image.
Yep.
Got it?
Yep.
Okay, next one. This one is a little tricky, right? Because there's like so much word to look at.
No, it was not tricky though. But like, you could understand like which is smaller and then bigger and then more bigger.
I think you thought it was going like smaller to more.
I didn't write the question though. I just answered.
This is the answer. Chloroplast, plant cell, leaf through
Rosebush.
rosebush. You got it. I trust in you. This one. If ribosomes of a cell were destroyed, what effect? We talked about this earlier. I think you like mistakenly. Yeah. And in the leaf of a plant, guard cells help to regulate. We mentioned this earlier, right?
Yeah.
About the stomata.
I don't even know why did they put that. There's like ultraviolet rays.
I know why. Okay. That's it. You got
5 wrong.
Yep. That's fine. Let's exit that. Let's go back.